right. So using bioengineering to make proteins, make products. How about gene therapy? Can we use biotechnology to alter a person's DNA within their body, right, as they're alive, etc.? Right now, not very well. We are still having a hard time with that. That's this idea of gene therapy. It says biotechnology can help diagnose and prevent diseases, but has had a limited success in curing them. It's pretty difficult to cure because you have a whole organism, and how do you fundamentally change their DNA? We could do genetic screening, absolutely. We can look at people's DNA and see, what versions of the gene do they have? What's the likelihood of having kids with certain diseases? We can do that. Will a baby be born with genetic disease, so cystic fibrosis, sickle cell anemia, Down syndrome, others? We could definitely answer those questions now, but we can't cure them. Okay, but we can tell people the likelihood of this or that or the other, and that way, if they're informed, they know what to do. Sometimes certain genetic diseases, all it takes is a little change in the diet, and that can help. Sometimes certain things that the mom does or the baby does, that can help. Is an individual likely to develop genetic disease later in life? Breast cancer, prostate cancer, skin cancer? Absolutely, we can look at those. And it's like, you might say, but I don't want to know. Ms. Collins, I don't want to know. But yeah, you do. Because if you know, then you might get checkups more often, more frequently. And if there's something different, you can diagnose it, get it out of the body quickly. And with cancer, for example, earlier detection is the best form of prevention, right? Because cancer is this thing where the cells spread, and if we can catch them before they spread, we can get rid of those mutated cells. So yes. Ethical dilemmas. Could there be discrimination? Health insurance, how to proceed with the information. For example, if people know your DNA and if they know that you carry these faulty genes, are they going to discriminate against you? Oh no, Ms. Collins, they don't do that. Good luck with that. Health insurance. Will they charge more for certain people with certain, genes? Will they discriminate, meaning they won't give you health insurance if you have certain genes? The jury is out, right? They already do it with people with pre-existing conditions. So, yeah. Here, this idea of trying to treat, trying to cure. This child has an immunodeficiency disease. And yeah, they have been successful with some kids, not successful with others in regards to helping their immune system. Gene therapy has had a poor record of success in curing diseases. Why? Because, it's difficult to fix the whole organism. Difficulty getting the working gene into specific cells where it's needed. Difficulty getting the working gene into enough cells at the right rate. Difficulty arising from transfer organism into unintended cells. Difficulty regulating gene expression. So when you're talking about the whole organism, it gets really difficult. So finally, in this chapter, there's cloning. Cloning, making exact copies. What? When I was in college, that was science fiction. Now, is it real? Can we do it? Yes, we can. By the way, I don't know if you knew this. You can clone your dog or your cat. Do you know that? For a few $100,000. Yes. I believe, it was a few years ago where I heard about it in the radio. I think it was a Korean company, but I'm sure more than just Korean companies do this, where yes, you can clone and make an exact copy of your dog. Now, is it going to be exactly your dog? Probably not. Because it's this idea of DNA, right? Nature versus nurture, how it grows up. And of course, there are many factors that influence an individual. So cloning, ranging from genes to organs to individuals, offers both promise and perils, right? How do they do it? So Dolly the sheep, that was a huge, it's more than 20 years ago now, where it's this mammal, it's this complex organism that was cloned. How did they do it? Okay. These are the steps. It says isolate an egg cell from one sheep and a mammary cell from another. Remove the nucleus from each cell. Okay, here's one sheep. Take a cell, remove the nucleus. Here's another sheep. Take a cell, remove the nucleus. Okay, so this is the egg cell, right? Egg cells, they could become embryos. This is mammary, right? Oops. The nucleus, remember, has all the DNA. So from this sheep, they took the nucleus with all the DNA, put it into this egg cell. Okay, so here's this egg cell from this donor. Here's this nucleus with the DNA from this sheep. Okay, so here's this egg cell with the DNA from this sheep now. They did some stuff to it. It says induce cell division, simulate normal early development. The cell replicate into 2 cells, 4 cells, 8, 16, 32, a ball of cells. Then they took this ball of cells that has the DNA from this sheep, implanted it into a surrogate mother sheep, and the baby grew, and then the baby was born. And if you look, it's white. It's identical to which parent? It's identical to this because it has the DNA from this sheep. So this sheep, this baby, is a clone of this, okay? This was a very famous picture on the cover of, I think it was Nature. So Dolly the sheep, right? She's identical to this sheep. Now we've done it with other organisms, cats, rabbits. Yeah, and we could say, why do we do it? Have we done it with humans? Oh my gosh, talk about science fiction, right? There have been a lot of movies that talk about this. Supposedly, no. In this country, there are laws against it. But you wonder, how about other countries? Could they be doing it in other countries? Kind of like Star Wars and the clone armies. It's difficult to do, and it took a really long time to do it. And the ethical considerations, for example, if this thing died, is that, let's say if we did it with humans and this thing died, is that abortion? I forget how many times it took. It took more than 200 times for this to actually work out. So you're killing a lot of embryos, a lot of organisms as you're doing this. So there are a lot of ethical issues. Plus, do we clone organs, or do we clone individuals to make organs for other people? I mean, what are the ethical dilemmas? You can see a lot of ethical dilemmas. But as of now, modern science can do this, and it's really wild. By the way, as humans, do we have any clones out there? Do we have any natural clones? Yes, we do. Who are they? Who are our natural clones? Identical twins. Identical twins are natural clones, okay? It would be a natural process where one egg, one sperm, fertilize, you make one cell, just like, this cell over here, but naturally. Then the ball of cells grows, and something happens naturally where the ball of cells splits and makes 2 individuals. So identical twins, not fraternal twins, identical twins, their DNA is identical. A little tidbit, are their fingerprints identical? No. What? They're identical. Their fingerprints aren't identical. No, they're not. In the uterus, in utero, when they're growing, there's pressure that is put on to our fingers as our skin is developing, etc. And so that's that nurture part, the environmental part. And one twin's fingerprints are going to be different than the other. But their DNA is identical. So that's kind of really cool. All right, let's see. Are there any medical justification? Oh, I thought we were done, Are there any medical justification for cloning? Can humans be cloned? Should they be? Yeah, we have to think about that. Finally, right, DNA and fingerprinting. Each person has their own unique DNA, except for identical twins. Identical twins, they are identical DNA. And we can compare DNA of one person and another. And they do this. That's like a fingerprint to identify a person. Let's say if there was a crime scene, and if you have DNA in your hair, in your saliva, milk, human milk has DNA, semen has DNA. Skin cells have DNA, depending on the skin cells, but blood, okay, there's DNA. So if there are any bodily fluids, it can compare the segments of the DNA and they can identify that person. And they do that for crimes, right? 99 DNA from different humans, 99.9 are identical. So what differs? There's these little 0.1% of our DNA that's different, and that's what they use to analyze. They're called STR, short tandem repeat. They look at that and compare them between one person and another to try to find out, did this DNA come from this person or that person? All right, you guys, you stuck with it. You've been strong. Okay, I'm going to have a quiz for this chapter. We're going to do a lot of activities in lab to solidify and review, especially with transcription and translation, because those are difficult concepts. Okay, hope you are well.